Third, activation of KRAS. Now KRAS became very popular. It's one of the targets of new drugs already existed in the market the last 1 or 2 years. And we know that KRAS regulates the growth factor of epidermal growth factor receptor stimulation. It's the first molecule of the cascade known as transduction pathways, known as RAS, RAF, MEK, ERK, which actually stimulates the nucleus. And then we have the loss of p53, extremely popular. It's actually the guardian of the cell cycle. p53 is a protein which form a tetramer and actually gives time to cell cycle to arrest in late G1 to S phase, as you may know by the cell cycle, and give time the cell to decide if it will go to cell death or it will repair the damage of DNA. So all these alterations leads to carcinoma, and if this carcinoma develops what we call metastatic phenotype, so it has the ability to invade and metastasize, then we will have formally full developed of cancer phenotype. So when we have what the clinician says borderline disease, the carcinoma in situ, then when they can develop the ability to invade the basal membrane and spread to distant organ, the cancer cells has to change their features. It's what we call epithelial to mesenchymal transition. It means that the cancer cells have to look like blood cells, or at least not to look like epithelial. So they hide or they downregulate all these membrane hallmarks so that they will sustain their presence inside the bloodstream. Those cells are circulated until they reach specific target organs. Nowadays we know that specific types of cancer metastasize to specific organs. For example, non-small cell lung carcinoma, they used to metastasize in suprarenal glands. We know that colon carcinoma metastasized formerly in liver and secondary lungs. So it means that there is some kind of seed and soil theory. So these are the circulated tumor cells. When they reach the target organ, they perform the opposite process called MET, mesenchymal to epithelial transition, and they engraft as micrometastasis, which is impossible to detect it with the compatible diagnostic methods, no matter how sensitive they may be, PET CTs or MRIs. And when they triggered, they start growing exponentially, and they provide you the macrometastasis, which can be detected. So, as you realize from that, circulated tumor cells have all the information which needed to provide distant metastases and recurrences of the disease. Also from clinical practice, it's very rare, almost never happens, that a patient dies from the primary tumor. Always dies from metastases and recurrences, which they are always resistant, always with highly mitotic rate, faster growing, and at the end it kills the patient. So why chemosensitivity testing? What happened with this? We know that the genotype, the genome information, provides all the necessary information of how the cell behaves, what we call phenotype. Also, we know that since we are not look like the same, we are different, this is caused by a feature called polymorphism. It means that the sequence of our genes is not completely identical. This polymorphism leads to a different quality and quantity of specific antigens. Those are features that can be used by cancer on their benefit. Phenotype is related from genotype, but also from the microenvironment, which means that if you take a tumor and you put it in a different microenvironment, it will have a completely different behavior. So it's not one-way system. It means that if you take a tumor and you change or you alter the microenvironment, you may have better or worse outcome. Also, you must have in mind that tumor is not one thing. It's a complex. It consists of multiple subpopulations of cells with different features and different abilities. So, when clinically we say that we remove a tumor or that we will have one tumor there, actually you may have 4, 5, 10, 100 different types of cells with different type of features. And finally, cancer cells are genetically unstable. Since the damage has been done in DNA, it has been a cascade of mutations and epigenetic phenomena which leads to genetic instability. And every generation of cell mitosis which is not normal in cancer cells, it means that we have new generations of cells with new features, new hallmarks, so they behave differently from the previous generation. So this number of subpopulation increase every time that we have mitosis. So it's extremely, therefore, to perform such kind of test so that we will know as many information we can concerning the abilities, the features, and the genetic profile of those cells. Let's see a few things about chemosensitivity. Actually, it was a necessity. It has been started by pharma industry because we know that the evaluation of new drugs performed in cancer cell lines at the beginning, and they need to develop methods so that they will calculate the response. This is the definition of chemosensitivity test. The ultimate purpose is to define which would be the response to a specific drug. Would it be cytotoxic,